Hello everyone, my name is Yash Agarwal. I am a second year MBBS student from NDMC Medical College, New Delhi. In this video, I'll be talking about translation. This topic is very much important for the NEET and 12th board examination. So without wasting time, let's move forward. Before talking about the definition of translation, let's recall what is central dogma. In my previous video, I have told that information that is coded in DNA is first converted into mRNA, and then mRNA is translated into protein or a polypeptide. So what is translation? Translation is the process of converting language of nucleic acid into language of amino acid. So the proper definition, which is given in your NCRT, states that it is a process of polymerization of amino acid to form a protein, right? So let's talk about when two amino acids are joined together, what type of bond is formed. So for example, this is amino acid 1 and this is amino acid 2. So when we have to join this, we have to remove a water molecule. So when a water molecule is removed, this process is known as dehydration. So to join two amino acid, dehydration is necessary. When water molecule is removed, this bond is formed. And this bond is known as peptide bond. This bond is known as peptide bond. Now you may be curious that why phenylalanine should come first or methionine should come first. How should we decide this? So what determines the sequence of amino acid? It is actually the sequence of mRNA which determines the sequence of amino acid. So this is an mRNA. One thing to note here is that translation occurs from 5' to 3' end. So this is our mRNA. The region flanked between the initiation codon and termination codon is known as translational unit. And this translational unit will form a polypeptide. This translational unit will form a polypeptide. So moving forward, let's talk about steps in translation. Translation is a very long process. But to shorten it, we will discuss it into 3 stages: the initiation stage, elongation stage, and termination stage. Before proceeding forward, let's understand the structure of tRNA. tRNA, or the adapter molecule, also known as soluble RNA, has a secondary structure of a clover leaf. But the actual structure of tRNA, that is the tertiary or the quaternary structure, is an inverted L shape. In this video, we will discuss about the clover leaf structure. So this is the clover leaf or the secondary structure of tRNA. Here we can appreciate 4 loops: the acceptor loop, T-C loop, anticodon loop, and T loop. In acceptor loop, we can see that 3' end is slightly longer than the 5' end. And at the 3' end, we have a specific sequence of CCA, that is cytosine, cytosine, adenine. To this sequence, a particular amino acid will bind. In T-C loop, the psi is a abnormal uracil. Psi means abnormal uracil. Here we can also appreciate a variable arm. In anticodon loop, we have a specific sequence which binds to a specific sequence on mRNA. This sequence determines which amino acid will attach at the 3' end. Hence the name anticodon loop. In the T loop, T is thymidine. So this loop will be rich in thymidine. Now we discussed that a particular amino acid will bind to 3' end. But this reaction is very high energy requiring process, and this also requires a enzyme. The name of the enzyme is amino acyl tRNA synthetase. What is the name? Amino acyl tRNA synthetase. So in this scenario, UAC will bind to AUG, and we know that AUG has two functions. It act as a initiation codon, and it also codes for methionine. So at 3' end, methionine will get attached. When tRNA is not attached to any amino acid, it is known as uncharged tRNA. It is known as uncharged tRNA. But when it binds to amino acid, it is known as charged tRNA. And this process is known as charging of tRNA or amino acylation. Amino acylation of tRNA. So now let's talk about the steps of translation in detail. We will keep prokaryotes in our mind, but there is no big difference between eukaryotic and prokaryotic translation. So the major difference we can see is a type of ribosome involved. In prokaryotic, 70S type ribosome is involved. So starting with the translation, this is the mRNA, which is formed from transcription. This is the initiation codon. And this, the red colored one, is the 30S subunit of ribosome. This 30S subunit of ribosome will come and attached near to the initiation codon. This process is aided by certain factors known as initiation factors, right? When this binding is complete, so the first tRNA carrying methionine will arrive at the P site. So this is very much important. It will arrive at the P site. In total, a ribosome has 3 sites: A site, P site, and the E site. Now, after the arrival of 30S, then 50S will arrive. It will bind to the 30S and will complete the whole structure of ribosome, right? So this whole process is known as initiation. This whole process is known as initiation, right? Now, at P site, there is tRNA carrying methionine, right? Now the next sequence is UAA. So tRNA, which carries amino acid phenylalanine, will arrive, right? Because UAA, according to the genetic code, codes for phenylalanine. So this tRNA, charged tRNA, will arrive at the A site. Then we have to form a bond between methionine and phenylalanine, right? So this process requires a peptidyl transferase activity. This process requires a peptidyl transferase activity. So this reaction is carried out by 23S rRNA, which is a ribozyme. What is ribozyme? It is RNA which functions as enzyme. It is RNA which function as enzyme. This is very much important. And where it is present? It is present on 50S subunit of ribosome, right? So this is the tRNA which is attached to the phenylalanine and methionine. So there is a formation of peptide bond here. Remember, this tRNA is at which site? A site. Now tRNA which is present on P site will shift to E site, and tRNA which is present on A site will shift on P site. So if we see like this, if this is A site, this is P site, and this is E site, what was present on A will shift to the P site, and what was present on P will shift on E site. So ultimately A site will be again empty, and it will be ready to accept another charged tRNA. So again another charged tRNA will come at A site, and the process will continue. Will this process continue forever? No. When the ribosome recognizes the termination codon, what are the termination codon? UAA, UAG, and UGA. When these codon are encountered, the translation is stopped. Does this stopping of translation requires any factor? Yes, it requires releasing factor, or the RF factor. And this RF factor results in dissociation of ribosome and release of polypeptide. So let's recall all the steps of translation. So first mRNA arrives, right? Then 30S subunit, or the smaller subunit, will bind near to the initiation codon, right? Then charged tRNA carrying methionine will arrive at P site. After the arrival of 30S, there will be arrival of 50S. Then 50S will bind to the 30S subunit, completing the structure of the ribosome. This process is under initiation. Then elongation happens. In elongation, we have to form a bond between methionine and phenylalanine, right? In this scenario, phenylalanine, right? So charged tRNA will carrying phenylalanine will arrive at which site? A site of ribosome, right? Now this peptide bond formation between methionine and phenylalanine is carried out by a ribozyme. Which ribozyme? 23S rRNA, which has a peptidyl transferase activity and is present on 50S subunit. This is very much important. It can directly be asked as a MCQ. Then there is a formation of peptide bond, and the you should remember that this tRNA is still present on A site, right? And the tRNA which was attached to the methionine is broken. Now shifting will take place. What was present in A site will shift to P site, and what was present on P site will shift on E site, right? Again, now A site is empty. Another tRNA, which will correspond to the sequence present on mRNA, will come and will continue the elongation process. Will this elongation process continue forever? No. When a releasing factor and the ribosome identify the termination codon, that is UAA, UAG, or UGA, the dissociation of ribosome and the release of polypeptide will occur, which will terminate the translation process. Now let's talk about high-yield information. Now we are student of medicine, right? So we have to learn about certain antibiotics. So we will discuss about 3 important antibiotics: streptomycin, tetracycline, and chloramphenicol. Now, streptomycin, how does this antibiotic act? This streptomycin will bind to the 30S subunit and will distort the structure of ribosome. Thus, there will be no protein formation, and thus the bacteria will eventually die. Another process. We have tetracycline, right? This tetracycline interferes the binding of charged tRNA to the A site. What does tetracycline do? It interferes the binding. It interferes the binding of tRNA to A site. Now, the last, chloramphenicol. The chloramphenicol inhibit the peptidyl transferase activity of 23S RNA. So, chloramphenicol. This is present in various cough syrup, right? So, this inhibits the activity of 23S RNA. Thus, there will be no polymerization occurring. Thus, the polypeptide will not be formed. So let's discuss about UTR. UTR is untranslated region. What is the definition? These are the region of mRNA which are not translated. It is present both at 5' end and 3' end. What is the function of UTR? It helps in efficient translation. Right? Let's discuss one UTR, that is Shine-Dalgarno sequence. Right? It is present at 5' end and it is a purine-rich region. Remember, when in initiation, 30S subunit binds near the initiation codon. That binding is regulated by this sequence. We should remember that, for example, this is the 30S subunit, right? This 30S subunit will have various small rRNAs. One of the rRNAs is 16S rRNA, right? This will have a complementary sequence to one of the UTRs, and thus it will help in positioning of ribosome at AUG codon. So Shine-Dalgarno sequence helps in positioning of ribosome at AUG codon. And one more important thing, the Shine-Dalgarno sequence is only found in prokaryote. It is only found in prokaryotes. Thank you for watching this video. If you like the video, please click on the thumbs up button, share this with your friend and subscribe the channel.